Hi everyone, Professor Gustin here, and I think you're going to find week number 9 challenging. In lecture, we're going to be doing microbial genetics, and there's some actually really cool things that I think you'll be able to relate to and can be meaningful to you with genetics. But I do know that students are challenged by DNA, RNA, proteins, transcription, translation, and I just want you to understand those basic genetic concepts enough that we can talk about some unique things in microbiology. So I'm not asking you to be a genetics and transcription translation expert in this, but please pay attention to the foundational information that I introduce at the beginning so you can understand the concepts about mutations and things that become kind of important and help you understand a little bit about the microbial world. So lecture should be, I think it should be good. I think it's actually pretty interesting. So hopefully you'll get a lot out of that. Lab, I would say this is the most challenging lab of the semester. I would not put it off to the last minute. Start working on your lab, because what ends up happening is there is some math in there. And there's dilutions, and what you're going to end up doing is you're going to try to count bacteria. When we say we're going to do an experiment that, actually one of them that we do is just how many cells are in one colony of E. coli. And how do we count that? How do you count millions of bacterial cells? So I'm going to show you how to do that, and we actually do this in the live lab. So there's a lot of things, a lot of examples you have to work your way through. There's a couple virtual labs that you'll be doing that will help you kind of repeat what you're doing in the handout. If you're having trouble with it, please let me know. That's why I'm asking you to try to do lab not at the last minute, but give yourself some time if you need to talk to me that you can. I did notice I did something weird in the syllabus. I'm not sure why. I had the lab listed as 20 points quiz and no virtual labs. But that isn't accurate. It's actually a 12-point quiz and 2 virtual labs, each for 4 points. So what you see on the Brightspace page is correct. The syllabus has just the content wrong, but not the total points wrong. So what do we have? So I would, we have our introduction right here. There's your lecture study guide and a bunch of lecture videos. I highly recommend that you use these lecture videos. The book has a lot of information on genetics and a lot that we couldn't possibly cover in a week. So I do this streamlining it and take out a few ideas that I think would be meaningful to you. So I highly recommend watching the lecture videos. There's a lecture assignment, just like we usually do, so get going on that. Notice that I have lab on here twice. One is a Word document and one is a PDF. There are some pictures that are really important for being able to do lab this week. And so some people's computers kind of mess them up. So I put a PDF document there. I know some of you like to type right into the lab handout. So you might want the Word document. But for most of you, probably the PDF document would be the better choice. These are identical, except that you can edit the Word document and you can't edit the PDF, and the formatting will be preserved. There's 2 virtual labs and a 12-point lab quiz. So get to it. Good luck, and definitely contact me if you need some help. Take care.